All right, guys. We continue our organic chemistry portion. So the next step is the protein. So we only have two more to go. It's protein and the nucleic acid. So protein is another very important organic compound or organic molecule. And so we need to just like the same pattern we learn for carbohydrates and also learn about the lipid. We want to know what is the monomer of the protein and what is the polymer. So just by looking at this primary structure of the protein, you can see that the protein structure is a linear structure. You can see that it has many like this green like dots right here. So each of these is a monomer. So a protein is a continuous string made by monomers, and each monomer is an amino acid. There are about 20 different type of amino acid, and the combination of those form the protein. So now we know the monomer of the protein is amino acid. So it's amino acid. The and the two amino acid bind together, and then three, and then four. Uh, Protein can be very long, and so we for a short length of the amino acids sequence, a chain, we call it a peptide. So peptide is a way to describe a short, maybe only like 3, 5, 7, 10. Those range are called peptide. And when it's getting longer, we call it polypeptide. And the longest one are called protein. When they become pretty long, it's not going to present as just a string. When they become pretty long, they will be folded. Our cell will fold it, use this long string to fold it. It's very much like knitting. So you have the yarn. This is yarn, and so you can knit that yarn to form a glove, and so glove can warm your like hands, right? A beanie, a vest, and so you can all of those can come from just a long yarn, and that's the same thing in our body. We use protein. We use uh, this polymer of amino acid, and then we fold them. We fold it. It's like knitting. In biology, we call it folding. So we fold them. So here I'm showing you the primary structure. This is the very beginning of the protein formation, uh, and it has the amino acids. Yeah, and then we can fold it. Cell will fold it from the. So this one is originally formed in ribosome. Ribosome convert the messenger RNA to protein, and then ribosome will transport this one into uh, endoplasmic reticulum and then transport to Golgi apparatus. And in the Golgi apparatus, they will be folded completely and also decorated. So here we have the primary structure, just linear structure. Then it can be folded. Primary just the sequence, the strain of longitudinal, the long uh, amino acid polymer uh, along this polypeptide. Yeah. So short one is peptide. Peptide usually we don't fold them because it's pretty short already. Polypeptide we start to fold them, and a long polypeptide is protein. Primary structure, linear structure. Secondary structure is a two-dimensional structure. So you can start with linear structure is a string, fold it, you can fold it. Let me show you here. Secondary structure right here. So you have the long A1, A2, that's amino acid, linear chain of amino acids. So that's the primary structure. Then you can fold it. You can fold it in a way like a coil. This is alpha helix. So that's way to describe that coil. Yeah. So these amino acids form different kind of intermolecular bond. So this is the weak bond. But with the weak bond, it will kind of like stick it together, fold together. So we can cells will fold it, use this kind of chemical attraction to modify the structure of a protein. So that's a secondary structure that is considered as a 2D structure. Now you can fold it further. Now you can see this one is called 3D structure. So it's not just alpha helix, but this alpha helix can further rotate it. And then, for example, like this one, it form a pocket. This pocket can help you to put your money inside, right? A little like pouch, put your money inside. So that's a tertiary structure. Tertiary structure, uh, one is linear, two is secondary, three is tertiary. So tertiary structure is a 3D structure. Then you can have many. You have one string folded to become a pocket, and then you have another one folded to become another component. And then if you put two or more peptide folded structure together, just like you have, for example, like this is what for example? Give me one example. Like all right, here you go. Let me find something. Let me find something for you to consider. Like maybe something like okay, like this is a pen, right? So pen has a two piece, like this piece. You can take it out, and then you put two pieces together. If you have two pieces together, it's called quaternary structure. Like this one. This one is a on the single peptide tertiary structure. Single peptide can go up to only tertiary structure. If you want to construct a quaternary structure, you need to have two or more peptide or amino acid polymer, yeah, together. So that's that. So here, for example, this is an example of hemoglobin. Hemoglobin is a typical structure. Hemoglobin contains four uh, uh, globular chain, four chain basically. This is one chain. This is a linear chain, right? Right here. What is that? Here, this is a chain. This is chain. And so where is that chain? Do we have chain here? See here. A sequence. Yeah. So here, linear chain. So here, linear chain. So one chain can be folded, and one chain can only go up to a tertiary structure. And the quaternary structure right here, you have one, two, three, four chain. So hemoglobin is a quaternary structure protein. It contains four chain, and these four of them basically are very similar to each other. Each one is like a pouch. Each one contains a coin, like this one here. In this case, it's a hint, but you can just think about it's just like a coin. Yeah. So that's a protein structure. So protein has to be folded, and the way that you fold it, it become a tool. So a glove is a tool, a beanie is a tool, hemoglobin is a tool. So example of this hemoglobin is that its function, so it's tool, so it has function. Its function is to carry oxygen. This one can carry one oxygen. Hemoglobin has four chains, four identical or four very similar things. It's not exactly identical, four very similar things. So each hemoglobin can hold four oxygen. Yeah, so its function is for oxygen transport inside of the blood. So protein is folded. So here you can have a folded, and then when we heat it, we will denaturize it. So we can denaturize it, meaning that we unfold it. So denaturation is a protein unfolding. Yeah, so you need to remember this terminology. Yeah, denaturation. Um, yeah, and you can fold it again, refold. Yeah. unfold, refold. Yeah. So heating can cause unfold. pH, heat or low pH, and both can unfold. So these give us idea that okay, protein when it folds, it's very tough to cut. Like you have a bundle of uh, wire bundled together. It's very difficult to cut it open, and so you when you hit it, you unfold it, and then it's easier for your enzyme to do the cutting. Cutting is catabolism. You break that you break that uh uh polymer apart, and when you put them together, it's called anabolism. So when you digest a protein, you heat it, so it's easier for you to digest. And in your stomach, your stomach has low pH. It can also unfold it, so for your stomach to digest it. Yeah. So that's that. So that's protein. Now the next part is the nucleic acid. So nucleic acid, the monomer nucleic acids. There are many type of nucleic acids. We have the deoxyribose nucleic acid, deoxyribonucleic deoxyribonucleic acid, or ribonucleic acid, ribonucleic acid. So we have different type of nucleic acid. What is their monomer? The monomer is called nucleotide. So that's this one. This is a monomer. Each nucleotide has three parts. You have the phosphate group, sugar group, and nitrogenous base. Nitrogenous base can have five different variations. That's how we store the genetic code. Yeah. So with this one is like just like a Lego piece, one attached to the other. When you attach to the sugar end right here. You attach to a sugar end. So this is where you attach to. Yeah, so let me show you here. So here, this one is a nucleotide right here. So you attach to this is sugar. This pentagon. This pentagon is a sugar. So you attach to it. So you attach to it. This is another one right here. That's another one. And you can see that this nitrogenous base is C, this nitrogenous base is G. And so this one just attached to it. This part can be varied, but this sugar and the phosphate are basically the same thing. So you attach to it, just like Lego, you attach to it right here. That's another one. All right. So that's another one. And that's another one right here. So yeah, so that's that. So here you have four pieces. So by here you can have the first one is C, second one is G, the next one is A, next one is U. This is an RNA. RNA you only have one strand. For DNA you have one, and you have the other one. So DNA you have two right here. So DNA you have one. So this one is sugar, right? This is sugar. So attached to a sugar, this one is a nucleotide right here. That is a nucleotide. And so attached to that sugar, another nucleotide right here. So 1, 2, 3, 4. And also here, four nucleotides attached to each other. On the other side, you have another one. So here is the nucleotide. That is the nucleotide. This one should be a phosphate right here. And the sugar attached to it. So that's another one. Right, that's another one right here. Not that, sorry. Not that, right here, like that. So this one attached to a sugar. Attached to a sugar. Like that. Okay. Oh, like that. So like that. Like that. So attached to it. And if you have two strands, that forms a DNA. And this one store, what's the major function of that? This one store the genetic information. GCAU, GCAT, yeah. So that's that. And that's for the organic finalize complete our lecture about the organic compound. I hope that's helpful, right? And the key thing, right? Let's go back. Let's go back. So what do you need to know? Quick review. So we now know what is the monomer of carbohydrate. That's the monosaccharide. Monomer of lipid, fatty acids, and cholesterol. Monomer of protein, amino acids. monomer of nucleic acid, that is a nucleotide. And this one, carbohydrate, lipid, protein, nucleotide, nucleic acids are polymer. So you can form monomer. We know they're monomer. Monomer uh, connect with monomer together, you form polymer. And so the formation of the polymer is through dehydration synthesis. Break it down is called hydrolysis. Uh, Forming polymer or like building a polymer is a catabolism. Sorry, is an anabolism. Break it down is a catabolism. Break down like eating or digesting, catabolism. All together is called metabolism. So that's everything. And then we know the feature of each type of organic molecule. The feature of carbohydrates, the feature of lipid, the feature of protein, and then the feature of nucleic acids. So that's that. All right. And thank you for, staying to the end, and see you next time. Bye bye. Sorry. Yeah.